Hello everybody, welcome to the third Revive webinar. I am Laura Piddock, Head of Scientific Affairs at the Global Antibiotic Research and Development Partnership, GARDP. For those who haven't joined our previous webinars, Revive is part of GARDP's Antimicrobial Memory Recovery and Exploratory Programme. It is an online space to connect and support the antimicrobial R&D community. It aims to facilitate learning, to connect people and share knowledge. The webinars are part of Revive's educational activities. In this webinar, we will have 4 questions which will be followed by 1 Q&A session. I apologise, 4 presentations, followed by 1 Q&A session. You can submit your questions at any time during the webinar, but please note that we will only discuss them after the final presentation. Please use the questions pane on your webinar control panel to submit questions. We will do our best to respond to as many questions as possible during the webinar time. Should we run out of time, please rest assured that we will make sure that your questions are answered afterwards, and we will make these available to everybody. Today's webinar is the second in the series, Clinical Development for Non-Developers. In the first part, David Shlaes talked about the traditional development pathways, Tier A and B. Today, David is back to discuss development of antibacterial drugs targeting specific pathogens. And he is joined by Friedland, Sumathi, Nambiar, and Mair, Powell. David has had a 30-year career in anti-infectives spanning academia and industry in the US, with a long-standing scientific and clinical interest in antimicrobial resistance. Having recently retired from anti-infectives in the pharmaceutical industry, he then had a consulting business himself. He now remains an editor for antimicrobial agents and chemotherapy, and as some of you are probably aware, is active writing a blog, Antibiotics, the Perfect Storm, and this includes in antibiotic policy areas. So before I hand over to David, I would like to take the opportunity to thank all speakers for joining us for this webinar.
Well, thank you very much, Laura. I'm very grateful to GARDP for its continued sponsorship of these sessions, which I consider to be really important. I'm also especially grateful to the distinguished panelists that we have today. I think it would be very difficult to get a more important and influential group of people to address this, what I think is a very challenging question that we're going to explore today. Freeland is one of the few clinical trialists really to have tackled this problem of developing antibacterial drugs for specific pathogens, and has had an extensive career in the development of antimicrobials. Dr. Sumathi Nambiar is one of the key decision makers and a leader at the Division of Anti-Infective Products of the FDA. And Dr. Mair Powell has been a leader also on the European regulatory side, both for the European Medicines Agency and MHRA. And I'm very grateful that they have all agreed to participate in this webinar today. Before we get started, I just want to acknowledge that this is September 11th. For those of you who are listening from the United States and around the world, it's an important day of remembrance for us. So just to recognize that. Getting on to our webinar, I have a few things I want to say before we kind of get started. What we're going to try and do is focus on these difficult pathogens, Pseudomonas and Acinetobacter today, as examples of the development of pathogen-specific antibiotics. And I don't think anyone should expect that we will actually have all the answers at the end of the day. And what this should tell you, those of you who are scientists working in the discovery area, what this should tell you is this is an area of higher risk than the kinds of products we discussed in the first webinar. This thinking about this webinar gave me an opportunity to reflect on kind of some what I think are bigger questions. One is, I mean, why do we perform clinical trials for antibiotics? Well, we want to understand how best to treat our patients, actually. And this includes considerations of both benefit and risk. We want to use the data that we garner in trials to show physicians and other stakeholders that our study drug provides an important alternative therapy for our patients. And as we discussed in the previous webinar, non-inferiority trials get us there because they show the drugs work. And this concept can be extended even to resistant infections. Even though data may not have been collected during the trial itself on such infections, we can use in vitro data and pharmacodynamic data to provide support for that conclusion. One of the questions we'll be talking about today is, are superiority trials actually superior? Another reason we perform trials is obviously we need marketing approval from regulators in order to make this drug, our new drug, available to physicians and patients. And we also want to convince payers that the drug under study is actually valuable. So it's a number of audiences that we're trying to address. I also was reflecting on the differences, if you like, between the way regulators think about things and the way clinicians think about things. Regulators approach clinical trials and medicines from the view of risk and benefit to a population. Clinicians really deal with patients one at a time. Each patient is different. A clinician sees each individual patient in the context of her or his experience and knowledge. And I might point out that this usually does not include the drug label, but usually does include knowledge of local conditions, sometimes literature, and certainly views of colleagues and opinion leaders. But a trialist, somebody who's designing trials, has to deal with all of these audiences plus the payers. Now, again, in reflecting, unlike some, I don't believe that regulatory requirements are currently a roadblock to antibacterial drug development. I just don't agree that that's true. I think the roadblock to further development of antibacterials, especially for small populations, is a community and a societal problem at this point, more than a regulatory problem, if you like. So again, pathogen-specific antibacterial drugs, specifically targeting Pseudomonas or Acinetobacter. To be clear, studying a broad spectrum agent as if it were a pathogen-specific drug is unnecessarily high risk, I believe. These should be developed using a more traditional tier A-B approach as discussed in the previous webinar. For a broad spectrum agent, if you wanted to demonstrate activity against these specific pathogens or resistant pathogens, a smaller study could be performed that would not necessarily be used to gain regulatory approval, but rather to increase physician confidence in the product. For these particular pathogens, the number of patients, as I'm going to show you shortly, the number of patients available for study decreases drastically, even when combining indications such as pneumonia, bacteremia, and urinary tract infections. If you only want to study those with resistant infections, these numbers become infinitesimally small. Again, to remind you, going back to the previous webinar from this wonderful concept developed by John Rex and his colleagues, traditional development tiers A and B include phase 3 studies. These are large studies, usually non-inferiority trials. In tier B, you might do one of those plus a smaller study. But with these pathogens, we're really often going to be talking about small studies or tier C because of the lack of available patients for study. And then, of course, there's the animal rule, which has been used mainly now for approval of drugs or other therapies for biothreat agents. So the problem is the numbers. And these data come from a presentation that John Rex gave at an FDA workshop back in 2016. And basically, if you look at the frequency of Pseudomonas aeruginosa causing infection in nosocomial pneumonia, it probably is on the order of 15%. For intra-abdominal infections, it's around 10%. For urinary tract infections, it's around 3%, and everything else is rare. So this constitutes a small number of patients available for study. I'll point out that Pseudomonas infections, from data that we gathered a few years ago, Pseudomonas infections are much more common, almost 10 times more common than those caused by Acinetobacter. But Pseudomonas is less likely to be carbapenem resistant, for example, than Acinetobacter. So if you're going to study Pseudomonas, you're going to be less likely to be studying highly resistant organisms. But if you're studying Acinetobacter infections, even though the numbers will be smaller, you're more likely to be studying highly resistant pathogens. So given the restricted numbers of subjects available for study, even when combining these indications, are non-inferiority trials feasible, even with a larger margin? So remember from the previous webinar, the larger the margin for a non-inferiority design, the lower the number of patients you need to enroll in the trial. And of course, even if you succeed in such a trial, you're still going to need to rely heavily on your in vitro data and pharmacodynamics to show clinicians how to use the drug and convince them, in fact, that it's a valuable drug to use. What is the role, if any, of historical or external controls to reduce the number of patients studied? Could one use such controls in a non-inferiority trial, or is this an approach mainly for superiority trials? I believe that you can actually gather external control data by looking at contemporary patients in hospitals, perhaps hospitals where you're going to do your studies, your trial, and you can, if necessary, even validate these by randomizing some patients in your trial and using that to validate the external controls that you're using. So I think that this is something that we should consider. And I think it might lead to feasible numbers. There are problems, big problems, with historical controls. And the big problem is that they frequently do not reflect control response levels you might obtain during a randomized trial. And this has been demonstrated in a number of different indications, including both HIV and Ebola. But on the other hand, external controls might be useful if a random trial is infeasible because of the rarity of the condition, such as what we may be talking about today. There must be sufficient experience to ensure that patients not receiving therapy will have a uniformly poor prognosis, which I think would be true. The therapy must not be expected to have substantial side effects. Of course, we need safety data for that. There must be a justifiable expectation that the potential benefit to the patient will be sufficiently large to make interpretation of the results of a non-randomized trial unambiguous. And finally, the scientific rationale for treatment must be sufficiently strong that a positive result would be widely expected. And here we would depend highly on in vitro data and PK/PD preclinically. And as I mentioned, you could try and validate your external control response rates using small numbers of patients randomized during the trial. So non-inferiority, even with embedded superiority, is the least risky approach. It does require larger patient numbers, and I'm not sure that this will ultimately be feasible for what we're talking about today. We have very little modern experience with superiority trials for antibacterials, but this is a standard approach in other therapeutic areas. Why do people object so much to superiority trials? Well, according to Drs. Rex and Talbot, certainly authorities, we must never knowingly randomize to ineffective therapy. Hence, routinely showing superiority requires that antibiotic resistance is so common that we can get sufficient numbers of patients into a randomized trial. And the best available standard of care therapy is in truth not very effective, which would be a situation, of course, we all want to avoid if possible. And then we show superiority based on excess deaths or excess morbidity due to the ineffective standard of care therapy in the comparator arm. And finally, once you show superiority, then that drug, the superior drug, becomes standard of care and must be the new comparator, right? No. And they are difficult, these trials are difficult to enroll. But getting back to the question of does a superior drug become standard of care, at least in the United States, the answer to that so far is a very clear and resounding no. These are number of cases treated. These are carbapenem-resistant Enterobacteriaceae infections treated in the United States since the launch of ceftazidime avibactam. And what you can see is the vast majority of these patients are still treated with colistin and polymyxin and not with a clearly superior drug. So standard of care has not changed yet. I'll also point out, and I'm sure you're going to hear this from our panelists, Europe has tools that the US doesn't. Conditional approval with re-review at a later time, approval under exceptional circumstances. They have both the US and the Europe would allow superiority trials and external controls, but as I mentioned, external controls are problematic. And with that, what I'd like to do is move on to Ian Freeland, who's going to talk from his vast experience on clinical development for pathogen-specific indications. Ian, I'm turning it over to you.
Hi, good morning, good afternoon. I'd like to first thank the organizers for inviting me. It's great to join David and our regulatory colleagues to discuss what's a very important topic. First, let me state that the opinions I'm expressing are my own. I'm not speaking on behalf of any specific pharma company or pharma in general. And I just want to make the disclaimer that if I occasionally sound bleak or discouraging, that is not my intention. My intention is to highlight some of the challenges and pitfalls in these kinds of pathways so that we don't underappreciate where mistakes can be made. Now, there are two FDA guidances, and Sumathi will talk on these as well. There are two guidances that specifically, I think, are relevant to this topic. The first is unmet need guidance, which has been around for a few years, but was finalized last year. And the main tenet of that is that it is appropriate to exercise the broadest flexibility in applying the statutory standards. And this guidance and the new statute has definitely led to major improvements in feasibility of clinical trials. The second guidance relates to LPAD, which is the Limited Population Antibacterial Drug, which came as part of the 21st Century Cures Act, which was passed in 2016. And this act was intended to permit the approval of new antibiotics on the basis of relatively small clinical data sets. And it does state that the FDA's determination of safety and effectiveness must reflect the benefit-risk profile of the antibiotic in the limited population, and this is my emphasis, and must take into account the severity, rarity, or prevalence of the infection the drug is intended to treat, and the availability or lack of alternative treatments for that limited population. So I think folks can try and interpret what exactly that means. But to this end, the FDA has a guidance which came out very recently, in which they remind us that substantial evidence of effectiveness for the drug's intended use and sufficient information to conclude that the drug is safe is still needed. They specifically state that this does not alter the approval standards, and the unmet need population is largely the same as what was described in unmet need guidance. A couple of other points to note is that it's not appropriate for drugs to follow this LPAD pathway if they can follow alternative pathways. This pathway can't be used to salvage failed drug trials, and the labeling should define the limited population. Comments that I have on this guidance, one is that it spends quite a bit of time stressing the labeling and promotion restrictions, but does not clarify how this pathway differs from the unmet need guidance in terms of data requirements, and in fact largely quotes the unmet need guidance. So it might be interesting in the discussion period to see if we can try and clarify how LPAD advances data requirements. The EMA guidance, which Mair Powell is going to speak in some detail, and as David pointed out, potentially has greater flexibility, but these larger flexibility elements are largely untested. So let's look at development for pathogen-specific agents. The good news is that regulatory agencies are really committed to trying to find solutions for development of agents for these pathways and to finding feasible solutions. They have, in my experience, met with sponsors very frequently, and they do provide very helpful advice. There are no recent examples of approval for Pseudomonas or Acinetobacter specific agents. So there haven't been any test runs of what an approvable data package would look like, and also importantly for the FDA, how the advisory committee would consider such data packages. It is important to note that good trial design comes with experience, and the chances of us getting the pathway correct the very first time we do it is challenging. And let us just consider how much trial designs have changed over the years for the common indications. How, for example, HAP/VAP guidance has changed from the very first guidance. Now, there are, as we know, many feasibility challenges, and this is not only in terms of the enrollment numbers, but I think it's very important to consider that sometimes these small trials introduce a number of confounders that make the interpretation of the data extremely difficult, and this is an area that really has to be considered very carefully. Many of the new therapies in the pipeline are non-traditional therapies, and these pose additional challenges. Although progress is being made, this is still early days. So what are the options for a pathogen-specific trial? One could do a randomized controlled trial at one infection site. This definitely leads to a much easier protocol and much easier interpretation. However, given the incidence of these kinds of infections, it may not be feasible to only conduct a trial at one infection site. And therefore, it is likely that we have to consider infections across multiple body sites. There is no doubt that this leads to a much more complicated design and interpretation. And in particular, combining different endpoints across infection types that have different severities is confounding and can be difficult. There can be additional confounders that are site-specific, and the PK may vary at different sites. Choosing comparators is always a challenge, but if one is looking at multiple body sites, this can be especially challenging, and best available therapy can be very difficult to interpret because in many instances it's very poorly effective. This may not be so much of a problem in a superiority trial, but I think definitely leads to problems with interpretation in a non-inferiority trial. Now, we hear a lot about the use and implementation of rapid diagnostic tests as aids to these kinds of a trial, and I think they can definitely speed up the diagnosis and improve enrollment in the trial, but of course they don't increase the incidence of these infections. Now, there are two options for enrolling these trials. The first would be to enroll patients empirically, so to have an empiric regimen that includes the test agent plus adjunctive therapy to cover all the potential pathogens for that infection. The major advantage of this is that one could start therapy very early, which potentially would lead to better outcomes and less use of confounding non-study therapy. course, the advantage would, the major disadvantage is that one would need to enroll many more patients. So for example, if the incidence of the pathogen was only 20% of that infection, which is typical of Pseudomonas in nosocomial pneumonia, one would need to enroll 5 times as many patients. This clearly is going to be a big problem, and therefore it is more likely that we would conduct trials where the pathogen is confirmed before starting specific therapy. The advantage, of course, here is that all patients would have the target pathogen, but the disadvantage would be that there would be a delay in diagnosis of that pathogen, and even longer if one needed to look at the specific susceptibility profile, resulting in larger use or longer use of confounding non-study therapy, and also potentially diminishing the treatment effect, which would be an important consideration in a superiority trial. Whatever trial design we look at, predicting enrollment and enrollment times can be extremely challenging, particularly if we're trying to target a specific rate of resistance or a specific incidence of resistant pathogens. This varies dramatically from time to time. Often there are outbreaks, and if one does on one's estimates during an outbreak, one might find a few months later that that outbreak has been controlled and that resistance pattern has largely disappeared. In the plasmomycin experience, which was enrolling specifically a study in patients with carbapenem-resistant Enterobacteriaceae, multiple hundreds of patients had to be screened to enroll the final 69 patients that were enrolled. The major reasons for screen failures were confirming the identity of the pathogen. Sometimes this took longer than the 72 hours that was allowed for a non-study empiric therapy. Other additional barriers include getting consent from very sick patients. I think polymicrobial infections are greatly underestimated. Infections such as nosocomial pneumonia are frequently polymicrobial. And then low Apache 2 scores or other kinds of severity scores are important, particularly in a superiority trial where one is targeting a specific outcome, and that if one enrolls patients of too low severity, this can dramatically impact those kinds of outcomes. And also, during the conduct of the trial, emergence to a comparator can occur. So some important consideration. I think we all agree, or we are all going to have to agree, that crucial evidence for activity against the target pathogens in this situation is going to have to come from non-clinical data, in particular microbiological and PK/PD investigations, because clinical trial data are going to be limited. And if one looks at like what's the feasibility, what are the sample sizes we're talking about, even making very liberal assumptions, such as we would have a 20% non-inferiority margin, 80% power, and even if we assumed that the new therapy is better than the comparator, so for example, a 35% mortality with a new agent versus 40% for the comparator, one would require 118 evaluable patients. And if we were not prepared to take the risk of assuming that the new therapy was better than the comparator, and we assumed that, say, there was a mortality of 35% for both treatments, the sample size would increase to 178. Now, this is not the final sample size. This is just the evaluable population. And the final sample size would require a much larger number, depending on what specifically one was targeting. If we were targeting, in that first example, usual resistance, which means all comers, we weren't particularly focusing on any particular resistance pattern, would be 140 patients. But if we wanted to target, say, multi-drug resistant pathogens, and if they accounted for 50% of all pathogens, that would increase the sample size to 240 or more. There are other, this is assuming a one-to-one randomization scheme. There are other randomization schemes, and external controls could reduce sample size, and hopefully we'll get a chance to discuss external controls in the discussion. And I think the EMA has a very nice statement that I think Mair Powell repeat, is that the sample size may be driven primarily by feasibility rather than doing these kinds of complicated calculations, i.e. something that doesn't take longer than 2 years. Now, there's the magic number of 300 as a minimum for a safety database that has been discussed previously. But the question is, in this situation, is it still reasonable to target a safety database of 300? Because you can see from my examples, getting to 300 patients is going to be a challenge. I do want to just say a word about confounders. This is very important to consider in these kinds of trials. And this is because confounders have a particularly big impact when one is doing small studies. This is especially important in non-inferiority trials, but one needs to consider these as well in superiority trials because it can reduce the treatment effect, which is a risk to the sponsor. And the kind of confounders one needs to consider is how sure are we of the diagnosis and the specific pathogen cause of the infection. This is especially a problem in nosocomial pneumonia. Prior and concomitant non-study antibiotics can be very confounding. Having polymicrobial infections, and I think what's also underappreciated is having new infections that occur either while you're on study treatment or just very shortly following study treatment. And again, these are common in hospital-acquired ventilator-associated pneumonias. Resistance to the comparator can be confounding, particularly if it changes over the course of the study. And that if one needs to use adjunctive therapies, overlapping spectrum of these adjunctive therapies is confounding. The word about regulatory harmonization, I think there's no doubt that the agencies have made much progress in this field, and particularly for these kinds of streamlined approaches. There's been, as I say, much progress, and there's a lot of agreement between FDA and EMA. However, we do need to watch out for some areas that still cause sponsors problems where there may be non-alignment. In particular, like what is exactly the size and the nature of the data package that would be acceptable, how exactly one defines the unmet need, and this is going to be a problem where the agent you are developing is targeting resistance in another class of drugs. The non-inferiority margins that would be acceptable and the alpha that would be acceptable for superiority trials. I think endpoints are still somewhat of a problem, particularly for nosocomial pneumonia and potentially bacteremia. And then the choice of the comparator and the dosing, sometimes there isn't agreement among the agencies as what's an acceptable comparator. So are Pseudomonas and Acinetobacter trials feasible? I believe we have proven that a CRE trial is currently not feasible. So the incidence of these pathogens would need to be at least 4 to 5 times more common than CRE to fall into the feasibility realm. So for Pseudomonas, I think if one was conducting a non-inferiority trial, an all-comer trial, in other words, usual resistance patterns, that does sound possible. I think if we were specifically focusing on multi-drug resistance or trying to conduct a superiority trial, that's going to be much more challenging. For Acinetobacter, given the incidence, even an all-comer non-inferiority trial could be challenging. And if one was looking to do a superiority trial or focus specifically on multi-drug resistance, I'm very doubtful that such a trial would be feasible. Although I do note that the AIDA trial, which was an academic trial, it enrolled predominantly carbapenem-resistant Acinetobacter, and that trial was completed. Just a quick word about post-approval considerations. I think we spend a lot of time thinking about and planning to get to approval, but there are challenges that occur after the drug's approved. For example, if we come up with a solution where there is a large post-approval commitment, that can be a problem for small companies who spend all their money on running the pivotal trials and then trying to launch the drug. I think we also have to think about promotional considerations where the drug is labeled not specifically for the target pathogens. And just a last word about breakpoints. These continue to be problematic for existing drug classes where the breakpoints for the new agents don't match those for older drugs in the class. And that's where I'm going to end this talk and hand over to Sumathi.
Okay, Ian, thanks very much. Sumathi, you're on.
Yep. Thank you, Dave. Would like to thank the organizers as well for the invitation to participate in today's webinar, and thank you all for joining the webinar as well. So I'm going to provide a regulatory perspective on development of antibacterial drugs that target specific pathogens and sort of build upon some of the themes that have already been laid out by David and Ian. So if a bacterial species that is being targeted for the development program is identified relatively frequently, then the trials are generally feasible. So for example, if one has a product that just targets Staphylococcus aureus, it's, one can do a study in acute bacterial skin and skin structure infections. Or similarly, if you have a product that targets Neisseria gonorrhea, then one can study gonococcal infections. However, if the drug that's being developed or the product that's being developed just targets a single bacterial species such as Pseudomonas or Acinetobacter that you've already heard about, and these organisms are identified infrequently in any particular infection type, trials become a lot more challenging. And so designing scientifically sound and feasible development programs for such drugs has been one of our areas of efforts in the recent years. So we do recognize the potential clinical utility of such products, and we also acknowledge that there are challenges in conducting clinical trials for therapies that target a single species that occur infrequently at any one body site. Patients who have infections due to such organisms tend to be sick, and hence there is an urgent need to start effective therapy. Because there is diagnostic uncertainty at the time of presentation, there's often a need to initiate empiric therapy. And Ian's already mentioned this, there's often use of pre-study and concomitant effective therapy in such patients. And also, unlike chronic conditions, it's very difficult to identify these patients ahead of time or maintain a registry of such patients. Just a word on unmet need programs in general. These programs tend to be much smaller, and hence there is greater uncertainty both with regard to benefit and risk. We think this is acceptable for life-threatening and severely debilitating illnesses where one does not have too many effective options. And this is consistent with our regulations. But it's very important to note that even for programs that are addressing an unmet need, the clinical trials must still meet the statutory standards for effectiveness as described in the Food, Drug and Cosmetic Act. It's very important because the clinical programs are fairly small that there is a thorough evaluation of in vitro activity and activity in relevant animal models of infection. And risks and benefits of such products will need to be communicated in labeling. So over the last few years, we've had several public meetings to discuss this topic. So in July of 2016, we had a 2-day workshop, and the second day of the workshop was dedicated to a discussion around developing antibacterial drugs that target a single species. In March of 2017, we had another workshop as a follow-on to the workshop in 2016 to discuss animal models of serious infections caused by these two organisms of interest. And then April of 2017, we held a meeting of the Antimicrobial Drugs Advisory Committee. And I've given you the links to each of these meetings. Slides and transcripts from these meetings should be available for your reference. So at the workshop in July, on the second day, we had a fairly extensive discussion about a hypothetical case of an antibacterial drug that had activity limited to Pseudomonas aeruginosa. We discussed potential clinical trial designs and did note that all of them had challenges and limitations. At the animal model workshop, there was discussion around the development programs for 2 investigational drugs that are currently in development, one that targets Pseudomonas aeruginosa only, and the second one that targets Acinetobacter baumannii only. There was discussion around the role of animal models, the key attributes of the animal models, and shortcomings of some of the currently used models. As a follow-up to these public meetings, we did have an open announcement for research studies, and we have funded 3 research projects, and you can find more information about these on our website. So at the advisory committee meeting in April of 2017, we discussed 2 key topics. One is, what would the development programs look like for single species specific antibacterial drugs when the organism is not commonly identified in any one infection? And then, should a clinical development program not be feasible or the data not be interpretable, what might be the role of animal models of infection be? So what do we think is achievable with such programs? As Ian has already mentioned, I think we acknowledge that a very limited clinical data package is potentially possible. The evidence of activity and efficacy in relevant animal models of infection will be needed, and there will also be need for robust PK/PD data package. And human safety information will be fairly limited, and we'll also have safety data from non-clinical studies. So the potential options, and I think this has been mentioned by both David and Ian, for clinical trial designs. The first one I'll touch upon is non-inferiority trials. We think non-inferiority trials is a feasible option. However, there is a likelihood that assessment of treatment effect could be greatly confounded. One reason for that would be the use of prior and concomitant effective therapies in a reasonable fraction of the patient population. We are willing to consider a single non-inferiority trial in this setting, using a wider NI margin than we would typically use for a standard development program. For example, for studies of hospital-acquired bacterial pneumonia or ventilator-associated bacterial pneumonia, we allow for a non-inferiority margin of 10% if you're pursuing a standard development program, and a wider margin of 12.5% for products that address an unmet need. For single species specific products, we are in fact willing to consider a NI margin which is equal to the estimated treatment effect, or M1. Alternatively, superiority trials are an option, and they're easy to interpret. They provide a clear finding of efficacy. It might be feasible to conduct for the first couple of products that are being developed, because it might be possible to demonstrate superiority over current standard of care. As standard of care changes, these trials will become infeasible and or unethical at the point when the new therapy replaces the less than adequate comparator treatment. And given the risks with superiority trial in general, we've seen that sponsors are not willing to design the trial to demonstrate superiority. So as activity and efficacy in relevant animal models of infection will play a very important role in these abbreviated development programs. It's very important that the effect be demonstrated in more than one animal species that is expected to react with a response that is predictive for humans. The animal models that are being studied should be relevant to the clinical condition being studied in humans. And ideally, the study endpoint in the animal models should be similar to the desired benefit in humans. So what could some potential outcomes of these programs be? And this is not an all-inclusive list, but sort of 4 potential scenarios that we have thought about. The best one certainly would be where a successful clinical trial was completed and either superiority or non-inferiority was demonstrated, and there are no major safety signals of concern. But it's also possible that the clinical trial results strongly suggest that there's a lack of beneficial effect with the test agent. A third possibility is that the trial was so confounded that the efficacy of the drug could not be discerned. And in this instance, evidence of efficacy might primarily come from animal models of infection. Another potential scenario is that although efficacy was demonstrated, there were significant safety concerns, so that the benefit risk assessment is not favorable. As I've already mentioned, safety of the product will be assessed in non-clinical studies, as you would do in any development program. And based on the signals that are identified, appropriate monitoring will be included in clinical trials. Ian mentioned the number of 300. I mean, that is the sort of the minimum number we require. Just want to clarify that these don't necessarily have to come only from the phase 3 program. We could use data from healthy volunteer study, but usually at the proposed duration, those numbers tend to be small. Certainly, we might ask for additional data if there is a safety signal identified. And for such products, given that the safety database is fairly small, there might be a need for additional safety data to be collected post-marketing. And it's very important that such products be used judiciously and the safety be closely monitored post-marketing as well. So with that, I'll stop and turn it back to you, Dave.
Okay, Sumathi, that's great. Thank you very much. Mair, can you go ahead?
Yes, thank you, David, and hello, everyone. What I'm going to talk about is to cover several scenarios, because as you'll hear, our recommendations are pretty much the same, depending on what sort of drug you've got and which pathogen you're aiming for, or pathogens. But before I start, we are in the process of rewriting our guidance, so I'll just bring to your attention that the two guidelines we currently have, which are on the screen, have actually already been combined, and we do have a draft which is expected to be released for consultation in November. And my presentation today entirely reflects the combined new text, so it's up to date, but at the moment you won't find this in the public domain. So I'm going to cover the various situations in which pathogen-specific indications can be granted in the EU, because, as I've said, what we're asking for is pretty much similar across these different scenarios. So in the presentation, I'm actually covering an agent with potential to address specific resistance problems. So it's eligible for a reduced clinical program and a pathogen-specific indication in patients with limited treatment options. I'm also dealing with agents, whether or not they address an unmet need, some may not, that have a very limited spectrum of activity, which may be a single species or genus, as I've said, with or without addressing a resistance problem. And an agent of whatever spectrum, but one that is proposed to treat a specific pathogen against which it's clearly not going to be possible to get enough cases to demonstrate efficacy in an infection site-specific trial, such as a very rare pathogen or a pathogen that can cause very unusual or rare infections. And these situations can apply to unlicensed agents, combinations of agents, licensed or unlicensed beta-lactams with licensed or unlicensed beta-lactamase inhibitors. So this is not the critical point. The critical point is what is the pathogen or pathogens that is being chased, with or without attendant multi-drug resistance problems giving rise to difficulties in finding active agents. So the common considerations for these settings are indeed that we consider in the EU that the really critical evidence for activity against the target pathogens is going to come from the microbiological data and from the PK/PD investigations. And I include in that everything from the non-clinical efficacy trials to in vitro PD models to clearly identifying the PK/PD index and PDT and then getting your PTA right. And we don't think that we're going to get robust and, well, certainly not definitive evidence from the clinical trials. Nevertheless, we still recommend at least one randomized controlled trial, no matter how small, no matter how difficult. Firstly, because randomization as a step is an extremely important issue for us in the EU. It also facilitates the assessment of safety. So I don't want us to lose sight of that today, because we seem to be very focused on efficacy. But randomization and a control group developed that way is very important for assessing safety. Now, as Ian has already read to you, the sample size may indeed be driven primarily by feasibility. And what we don't want is trials that drag on for years and years and years and start to lose their relevance to current clinical practice. And we would think that approximately 2 years is the upper limit for continuing enrolment. The statistical analysis plan, accepting that this is not going to be fully powered and designed to address specific statistical issues, should address the performance characteristics of the trial. And then the considerations for a non-inferiority margin include ruling out what is considered or could be agreed to be a clinically important difference between test and comparator. Now, if the agent does happen to address also or only specific resistance problems, then in the EU, the critical issue is to establish eligibility, which initially would be done from the in vitro studies, and then, of course, from PK and PK/PD analyses. And we do have separate guidance in the EU on these issues that should be followed. We also point out that occasionally a pathogen-specific indication may still have to be qualified by the infection site. So you may have, for example, an agent that's active against acylnitrobamine complex, but the PK data is telling you either no active drug gets into the urine, which raises a question about using it in complicated UTI, or maybe the ELF penetration is extremely low, raising questions about HAP-VAP. And you're not going to be able to answer those questions from definitive, robust clinical trials. So it may be that a pathogen-specific indication is given, but it is additionally qualified in some way regarding the site of infection at which that agent is likely to be active. Now, again, as I've said, we do ask for one randomized controlled trial. If the spectrum allows, we recommend that it is in a single infection site-specific indication. And enrichment for target-resistant pathogens isn't required, although we do encourage people to think about that if a suitable licensed comparator becomes available. It does not have to demonstrate non-inferiority using recommended margins for standard indications, such as treatment of complicated UTI, if the trial is only intended to support a pathogen-specific indication in patients with limited treatment options. Obviously, if the sponsor wants to use the same trial to support a standard indication, then it would also have to meet the usual margin. For agents that address specific pathogens, whether or not they address multi-drug resistant pathogens, it is preferred still, as I've said, that we have at least one randomized controlled trial, if possible in one infection site-specific indication, and we strongly encourage sponsors to use rapid diagnostic tests to improve the feasibility of these trials. We do here say that it could be justifiable to conduct a trial that enrolls patients with infections at different body sites. Generally, we try not to accept such trials, but we recognize that very occasionally this may be the only way to accumulate sufficient cases and to accumulate sufficient safety data that we could consider giving an approval. To stress also, of course, that these trials are important because they provide PK data, and those PK data can be used to update the POP-PK model and re-estimate the PTA, and then it becomes relevant to very sick patients. Alternatively, or in addition, for some relatively rare pathogens, we recognize that sometimes you can actually also get some information from patient subsets. So, for example, if you have a drug that is aimed at or has very good activity against Legionella, you might be able to pick up some information on efficacy from small subsets that just happen to be enrolled into community-acquired pneumonia trials. So to summarize, a pathogen-specific indication, whether it's a species, a genus, or sometimes even as broad as aerobic gram-negative organisms, is possible in some circumstances in the EU. But it's very important that the eligibility of a product for such an indication is discussed with us early in development. Our revised guidance, hopefully to be available quite soon to you, aims to provide a lot more clarity on the development of antibacterial agents that can address specific resistance problems and or treat rare pathogens. We do have guidance in our current documents, but it has become very clear in the last 5 years from our interactions with companies that they haven't always understood what it is we want. Furthermore, we laid out options in that guidance that in reality is not what we've been saying to companies. And so we are now trying to address these issues to make it very clear what we want, to stress that whenever possible, we do want at least one randomized controlled trial, regardless how small, regardless of how difficult to interpret, because we consider the randomization step to be very important. Obviously, whenever possible, whenever the spectrum of the agent allows, or the pathogen allows, then in a single type of infection and using a single comparator, but fully recognizing that there will be situations in which that is not possible. The RCTs will of course be limited in size, and it's essential therefore that we have very robust microbiology and PK/PD analyses to support these very limited clinical data. So I hope I've kept to my time. Thank you, David. Thank you all.
Well, that was great, Mair, and everybody else. Thank you very much. I'm already getting a few questions in. So I wanted to start with a few ideas that have come from some of the prior workshops, both in Europe and in the US. And one is the idea, especially considering, for example, a Pseudomonas trial, of pre-enrollment. So the idea would be that when a patient comes into the intensive care unit, for example, on a respirator, especially if you want to choose those patients who are already colonized with, say, Pseudomonas, that is, they would be at higher risk of getting a Pseudomonas respiratory tract infection at that point. Getting informed consent prior to them developing their pneumonia, and therefore being able to move a lot more quickly to get them onto a study. Do you think, Ian and Mair and Sumathi, do you think that will improve feasibility of such a trial?
Yes, maybe I can go first. I'm not a trialist, so we'd have to ask trialists, but we have discussed this with some clinicians, and it does sound like there is some feasibility associated with this, that there are other fields in which pre-consent is obtained. So I think that this could help. The one thing it doesn't really do is change the time it takes to confirm the diagnosis. So I think it would help in trials where one is doing empiric enrollment. But if we still need to confirm the pathogen, it still takes 3 days. So I'm not quite sure that it would absolutely help in that situation.
Okay. Sumathi and Mair, do you have an idea on that idea?
Yeah, David, this is Sumathi. So I know CTTI had done a project looking into this very specific issue, and I think the overall assessment is that it can certainly help. I don't think it'll solve all the problems, but I think the difficulty is with getting informed consent in this very sick patient population. I think it helps with that step. So we certainly think that if people are interested in pursuing that option, we think it's a good idea. But again, it might not remedy all the difficulties.
Right. Anything to add, Mair?
No, I don't think so. In principle, I don't think there's any regulatory objection to that approach. But I'm, along with Ian, I'm not sure how much it will help.
Yeah. Another question that came up is in terms of feasible non-inferiority margins. So what is it? For example, take Pseudomonas, which I think is the easier example here. What is a feasible non-inferiority margin, and what's the clinical importance of that margin? And I think this is mainly for Mair and Sumathi to try and grapple with.
Sure. I can start, and then Mair can add to it as well. So I think, as I mentioned in my during my presentation, I think we are exercising maximum flexibility, and as with any non-inferiority trial, we first have to quantify the M1 or the treatment effect, and then the question really is how much of that is one willing to give up in the trial that one is conducting. And I think purely from a practical standpoint, for feasibility reasons, for drugs that just target, say, Pseudomonas, we're willing to be fairly generous and use the entire M1 as the M2, and that's why we have agreed to a 20% non-inferiority margin. And from what we've heard in discussions with sponsors, I mean that it is sort of a stretch. I mean, it's going to be difficult. It's not easy to get 300 patients with just Pseudomonas infections who have HAP-VAP, but I think that's a reasonable starting point. Now, beyond that, I think if you try to extend the margin beyond that, then I think you run into the issue of not having a scientifically justified non-inferiority margin. So that, I think, for now is about the extent of where we would go, and I look forward to hearing from Mair.
Yeah, Mair, what's your thought on that?
I don't think I really got much to add. I agree with everything Sumathi said. I would only say that we like to look at these things very much on a case-by-case basis in the EU. So it would also depend a lot on how big we felt the unmet need was and how convinced we were that the drug is going to address it. But yeah, in principle, I agree with everything she said.
Okay, good. Let's see. One of the questions that came in was, given the difficulties of an Acinetobacter trial, and it sounds like in Europe this would not fly. The questioner is asking, could we simply use the animal rule, if you like, for an Acinetobacter trial? Because I think that's going to be about 10 times more challenging than a Pseudomonas trial.
Maybe I can go first on this one then. I don't necessarily agree with the person who posed this question, because our most recent experience is that, okay, not huge, but reasonable trials in Acinetobacter are possible. It may be necessary, and we could accept looking at more than one body site. But I don't think there is a need to resort to only non-clinical efficacy data here. I think something is possible.
Okay. Sumathi, you have anything to add to that?
No, I would agree with what Mair has said, and really use of the animal rule is for very select situations, and where really human efficacy studies cannot be conducted because it's unethical or field trials have not been feasible. And I think it was discussed at great length during the workshop we had in 2017. The animal models are not quite there yet for us to be able to feel comfortable to assess efficacy and then have the product available for human use. So I think at this point, we think trials are feasible. There will be certainly more uncertainty in these trials, so programs will be smaller. And as Mair said, we're willing to look at more than one body site, but I think attempting to do a clinical trial and getting some data is probably the best option at this point.
Right. So turning to randomized clinical trials, and especially to the point of view of Europe, what do you think about using external controls to boost the control population, but at the same time randomizing a small number to say standard of care or best available therapy or whatever, to provide a validation set for the external controls? This is something that I've been talking about for a long time, but the Infectious Disease Society has raised this as well. What would be the European view of that?
I wouldn't close the door to that, but I would have to be honest and say it is a long way from our preferred approach. We really don't like external controls at all. We would much rather that you have a very unbalanced randomization, even if it's 3 or 4 to 1, so that the control group is small. But it's very, very hard to justify the relevance of external controls unless they truly are parallel time frame and all sorts of other things. So I'm not shutting the door, but I would reiterate that it isn't what we'd really like to see.
Okay. Any thoughts from FDA?
So I would agree with Mair, and actually this option is included in our unmet need guidance, one of the guidances that Ian referred to. But just as Mair said, we are not comfortable with historical controls. External controls really should be reserved for special circumstances. And I think, David, in your slides, you had highlighted that, disease has to have a high and predictable mortality. Effect of the drug should be self-evident. But in these kinds of infections, you see that the outcomes are highly variable, and exactly how to borrow the information from historic controls is not very clear. I suppose there could be statistical ways of doing it. I'm not a statistician, so I don't want to go into that, but we have reservations. But if there is a very sound way that you can convince us that a lot of the biases that we're worried about are not an issue, we're certainly willing to take a look at it.
Okay. Another question that's come up is, okay, so if the animal data for Acinetobacter is not predictive, how do you come up with a robust PK/PD package? I think, Sumathi, that's you.
Yeah, so I think there's two things, right? I mean, I think there are uncertainties with the appropriate animal models for PK/PD, but I think what we were also referring to is if you want to use the animal rule and use that as the basis for approval, I think the animal models of infection we currently have are in no way, definitely reflective of human disease. And so our hope is that, we have funded a few projects. When those studies are completed, we hope we will learn from them. And I don't know if we'll have all the answers to the questions. Maybe we'll need to do further work. That's why we think the ongoing work on advancing the science of these animal models is very critical to help all of us move the field forward.
Okay, thanks. So one question I've been dying to ask all of you is, what do you see, given everything that's going on and there are a number of products now that are being developed or certainly are in preclinical stages approaching clinical development stage. What do you see as a feasible approach for example, in the worst possible case, an Acinetobacter product? What is a feasible approach? And I'd like to start with Ian. I mean, Ian, what do you think is a feasible approach from your point of view?
All right, so you're definitely asking the tough question. Pseudomonas, I could see a pathway. Acinetobacter to me is much more challenging. I think the nature of the disease itself is challenging in that the outcome, the treatment effect and the outcome seems to vary, and there are definitely differing opinions on the pathogenicity of Acinetobacter. So it's not just the incidence. I think it's also, trying to predict the outcomes and the attributable outcome from the Acinetobacter. So I really struggle with that kind of trial. I think it would have to be, in my opinion, a superiority type study, and that definitely introduces a large number of challenges. In particular, one would need to ensure a high enough negative outcome. So if mortality was the end point, it would have to be a high enough mortality in the control group that one could show superiority. I just struggled a little bit with doing non-inferiority trials because it's not quite clear what the treatment effect is of standard therapy at this stage.
Okay. Sumathi?
Yes, I think one difference between Acinetobacter and Pseudomonas, and I think, David, you mentioned this during the introduction, is the high prevalence of carbapenem resistance. So there might be an opportunity to demonstrate superiority here. The other thing we've discussed is, potentially going to geographic areas, because there are certain geographic areas where the prevalence of Acinetobacter infections is much higher. So targeting those sites might be another option. And certainly, as Mair had noted, including more than one body site. So potentially combining, say, HAPVAP and bacteremia in one study and using an outcome such as mortality is another way to potentially make the trial feasible. So those are some thoughts.
Mair, do you have any ideas?
I don't think I have anything to add. I continue to believe that a small trial that allows more than one body site infection in with Acinetobacter is actually still feasible. It's not going to be easy, but I do think that something can be done clinically. And as I said earlier, I don't see that in this particular example, we could or should resort to basing everything on non-clinical efficacy data.
Okay. So one of the questions that's come in is, do expectations for target attainment, predicted target attainment, increase or decrease for rare or infrequent pathogens or highly resistant pathogens? So in terms of making the target attainment level 90% versus 95% versus 99%. Mair, do you want to try for that one?
The short answer is no. On the other hand, if you read our PK/PD guideline, we do discuss scenarios in which we might be willing to accept that PTA is slightly lower than we would normally like to see. But it's not necessarily tied to these situations. It's more a consideration of the life-threatening nature of what it is you intend to treat or expect to be treating and things like that. I mean, yes, we can have some flexibility here too. We don't have a guideline that says thou shalt and must always meet X% PTA, but we do discuss the scenarios in our guidance.
Yeah. You have anything to add, Sumathi?
Nothing, Dave. Thank you.
Okay. So Sumathi, one for you is, what are actually the main practical differences between LPAD and unmet need?
Ian, I guess I need to thank you for bringing that up. This is very complicated. Let me try to explain in sort of simple terms. And just to let you know, the LPAD guidance that Ian referred to, is a draft guidance, and that has been posted for public comment. So if you have any questions or comments, here's the opportunity to comment on it. So really, LPAD is what the LPAD gives us is really a tool, and there are sort of three basic requirements for LPAD for a drug to be considered under the LPAD pathway. First is it should be intended to treat a serious or life-threatening infection in a limited patient population. And very importantly, and Ian mentioned this in his slides, it does not change our standards for approval. So even if a drug is being considered under the LPAD pathway, our standards for approval, our evidentiary standards, do not change. And then what we also need is a written request from the sponsor to approve the drug as a limited population. So the main thing that LPAD gives us is that, you know, the risks and benefits are acceptable in a very well-defined patient population. And this patient population that we define should make clinical sense. So, you know, a practitioner should be able to identify who is the patient population for whom the FDA has determined that the benefits outweigh risks. So that's a definition of the limited population. And then really a lot of it relates to labeling and the need for promotional materials to be submitted before dissemination. The standards for approval don't change. The clinical trial design options, and that's why there's a lot of reference in the LPAD guidance to the unmet need guidance, because many of the development programs outlined in the unmet need guidance are still relevant. So we do think that of products that are developed that target a single species, because the programs are going to be so small and you can define the patient population, they might fit under the LPAD pathway, but you know, we don't know. These products are still in development. So after the clinical programs, I mean, they're done with the clinical trials, we'll have to make that assessment. So I know this is a little confusing, but there is a fair bit of overlap, but I think the main difference is in defining the limited population. But whether it's unmet need or LPAD pathway, you have to meet the evidentiary standards. And I think there's a little bit of misunderstanding that the evidentiary standards have somehow been changed because of the availability of LPAD.
Okay, well, I think that helps. So, well, let's see. I'm getting several questions here. Okay, well, first of all, one person is asking, can a new drug class that was broad spectrum, but is also active against carbapenem-resistant Pseudomonas, be developed using the unmet need LPAD pathway? Of course, my first question to this person is, why would you do that? But if they wanted to do it, would these pathways, these unmet need pathways, be available for them? Mair?
I'm not sure what we're talking about here. I'm very sorry, but are we talking about a broad spectrum drug that someone chooses to develop only for Pseudomonas? I'm a bit unclear.
Yeah, it's a broad spectrum drug that they choose to develop to treat carbapenem-resistant Pseudomonas infections.
That would be very strange, as you say.
Right.
If it had particular benefits for Pseudomonas and it was active against carbapenem-resistant Pseudomonas, then I suppose they could do that, yes. But I think we would have to sit down and talk to them about this, because it would be an unexpected scenario.
Yeah, and like I said, why would you do that?
Yeah, I've not had anyone suggest anything like that to me so far anyway.
Yeah. Sumathi, anything to add there?
No, I think, David, you'd made the point in your presentation as well. If you have a broad spectrum agent, then just designing it for Pseudomonas sort of doesn't make sense.
Right. Okay, one question here is, at what stage of development is engagement with regulators recommended? During preclinical development, during lead optimization, how do we identify whom to contact? And actually, there are differences here between the US and Europe. So Sumathi, why don't you take this first, and then we'll go to Mair to tell people how to do this.
Sure. So, I mean, I think the key message from us is that we encourage frequent interactions, and you're welcome to come discuss with us at any stage in development when you're ready. We have a pre-IND consultation process when you're very early in development, and you know, that's an opportunity to get feedback on your non-clinical program. And more often, we are also discussing the overall development program, including what your phase 3 trials might look like, albeit at a high level during the pre-IND consultation process. And then as development proceeds, there are, certainly the milestone meetings, but other than that, we are more than willing to have discussions, meet in person, have teleconferences, or provide advice in writing. So I think we do encourage folks to come meet with us or talk to us as often as needed.
Yeah, I can vouch for the fact that these interactions with FDA are easy to do and extremely helpful. Mair, do you want to talk about Europe?
Sure. Just as in the US, we do encourage people to interact with us frequently. It's much less sort of formulated as to, I mean, we don't have an NDA, we don't have an IND process, as you know. So it's a little different, but people are welcome to come, even if they've just got microbiology data and think we may have something very promising here. We'd like to scope out with the regulators where we might end up clinically to maybe guess as to what the final development costs might be. So, you know, we're happy to talk to people really early if they want that, and then they can come back as they need. What is different, as you say, David, is how you do it, because you're not just dealing with with one regulatory body as you are with FDA. And in Europe, you have a choice. You can do one or both of talking to individual national regulatory agencies that you think have particular interest in the type of product you have, or you can go for CHMP advice, and quite a lot of companies choose to do both. They do the one first, put together a list of questions, and then they go for CHMP advice to get a consolidated EU opinion on the most important issues for them to address.
Yeah, thanks. I think my experience is going to an individual regulatory agency first is easier. MHRA has been very good that way. And then going to CHMP at a later stage seems to work well, as you say. So, okay, so I'm going to do, let's see, well, two more quick things because we're running out of time here. So Sumathi, one question is, given that the approval standard is the same under unmet need guidance and LPAD requires labeling restrictions and promotional requirements, why would any sponsor apply under LPAD? What is the quid pro quo for restrictions? And actually, I don't know that there is any difference in labeling and promotional requirements between unmet needs and LPAD, is there?
Yeah, so there are some specific labeling requirements for LPAD, and our guidance outlines all that. There's particular requirements for labeling and also for the carton and container. But you know, one can envision a scenario where safety and efficacy has only, you know, the risk benefit is only favorable in a very well defined, a very specific patient population. So in that instance, it makes sense then to have it under the LPAD pathway. And sometimes it could be you might have seen a treatment effect in a larger population, but that might be a subgroup in whom the risks and benefits justify moving the product, making the product available. So these, you know, we really require a fair bit of discussion with the sponsors. I think it's hard for me to give an answer that will suit everybody, but a lot really depends on the product, what the risks are, what the benefits are, and what the overall development program looks like. But bottom line is, can you define a population that makes sense clinically in whom the risks and benefits have been assessed?
Okay. Okay, I'm just going to do one more here quickly, and that is a question. Does either FDA or EMA have a list available of unmet needs for anti-infectives? And I believe in both guidances, there are examples of what might be unmet needs. There's also the CDC and WHO priority lists for pathogens and resistant pathogens. Do any of you have anything to add to that quick answer?
Nothing from me.
We don't have a list or examples in our new guidance.
Okay. There you go. I think we're going to stop here so that Laura can kind of wind up. Again, I want to thank all of you for participating. I hope to be able to call on you again in the near future. And I want to thank again Laura and GARDP for sponsoring these webinars. Laura?
Yes, so thank you, David. So I'm very sorry, everybody, that we have run out of time. We will post all the questions that we did not have time to discuss today. They'll be on the public forum on the Revive website in the very near future. So please make sure to visit revive.gardp.org to find the recordings of this and previous webinars, plus conference sessions, announcements of future webinars, as well as the public forum and talk to an expert feature. I would also like to thank David, Ian, Sumathi and Mair for this very valuable webinar. It's been fabulous, and thank you so much for such clear talks and responses to the questions. Last but not least, I thank everybody for joining this webinar and for contributing to the discussion. Goodbye.
Bye, everybody. Thank you.
Thank you.
All right.